This presentation is part of the online masterclass given by Professor Juri Kropotov as a complementary introduction to his new book, Functional Neuromarkers for Psychiatry. Professor Kropotov is a well-known neuroscientist from the Human Brain Institute in St. Petersburg, Russia. Professor Kropotov has developed a unique methodology for investigating cognitive processes in the brain using event-related potentials. This methodology includes a normative database and advanced software for computing independent components of ERPs. Please enjoy this presentation.
About functional neuromarkers in healthy brain. What kind of scientific facts we have now? Maybe some just few words about what we are measuring. I think that probably most of you know this, but just again, to give you a kind of an overview of what should I say this. So, as you know, the brain consists of little gray cells that are interconnected with each other. If you implant electrodes into a single cell, you can record impulse activity. So the neurons generate spikes that are traveling by axons to other neurons. And when they travel to other neurons, they generate the currents, also seen as potentials, which are called excitatory or inhibitory postsynaptic potential, depending on the synapse, if it's excitatory or it's inhibitory. As you know, in the brain, excitation and inhibition must be both present and must be balanced. And but if you implant macroelectrodes in the cortex, then you can record local field potentials, which we did in our studies when we implanted electrodes into clinical in psychosurgery. And you also can record the EEG and also event-related potentials from the skull. So just simply placing metal electrodes on the skull, you can measure this activity. Of course, the amplitude of this activity is much smaller than the local field potential, and of course it's much slower than the potential that are recorded by micro electrodes. But the beauty of this is that using new technology, new advanced method of EEG and ERP analysis, you can extract from this signal a lot of information, and you can use this information in clinical practice. So basically, by looking at EEG by naked eyes, it's almost impossible to say anything except seeing these signs of epileptic focuses. And as you know, that EEG in neurological clinics is used mostly for epilepsy. Okay, so this you know better than me, that there is a scheme of recording EEG from the head. And here I'm presenting a kind of a working space of the 21st century psychiatrist, which includes the amplifiers for recording EEG, the computers for presenting stimulus, computers for EEG recording. The psychiatrist actually usually don't have enough time for doing analysis by himself, except some advanced psychiatrists, but he usually sends the EEG for analysis to some center for specialists who do report on functional neuromarkers, and on the basis of this report, the psychiatrists make decision how, first of all, decision what is wrong in the patient brain, what is the dysfunction that would correspond to his behavioral pattern, and how this dysfunction can be treated. Okay, so this is the next slide in which I show EEG. So on the left, that EEG recorded in the referential montage. You don't see much, and just up and down, and some artifact. In this case, I blink. Artifacts, just to get rid of the common source, you can use the common average montage. Again, it tells you a little bit more, but not much. Using current source density, so instead of presenting potential, which are volume conducted, you can use the current source density, which actually gives you an idea of local currents in the cortex. That gives you information about local events. In this case, you can see quite clear the frontal midline theta, burst of frontal midline theta, burst of the parietal alpha. Okay, so what I'm trying to show on this slide is that even using different montages, you can obtain more information about brain function, but definitely not just looking at EEG. So you need to make some complicated, use some sophisticated method for extracting this important information from the brain. Can I ask, this slide represent, I think that it's kind of a knowledge about neuronal mechanism of generation of the rhythm that you can observe in healthy brain. Now I am talking about a healthy brain. And on the top, you see a kind of schematic presentation of mechanism generation of alpha rhythmicities. And we know that they are generated by thalamocortical networks, inhibitory and excitatory neurons in this thalamocortical network. We know that it's a family of alpha rhythm, which includes mu rhythm recorded from C3, C4, parietal alpha rhythm, and occipital alpha rhythm. And alpha rhythm, they actually idling rhythm of the brain, and from functional point of view, they show inhibition or closing sensory gate to the cortex. As you know, the thalamus is the gate to the cortex. When you close this gate, the thalamocortical network starts generated alpha rhythm. And the next rhythm that we know, and we know the neuronal mechanism for this rhythm generation, the rhythm is generated in this in the PAPI circuit, the circuit that includes the anterior cingulate cortex, the hippocampus, and quite complicated network. And the rhythm, the frontal midline theta rhythm, the only theta rhythm in the healthy brain, and it's appears in burst of quite short burst, and from functional meaning, it's open gates to the hippocampus. Then we also had beta rhythm. There is a family of beta rhythm, and different beta rhythm, they reflect different mechanism. We have, for example, Rolandic beta rhythm, which reflect the the functioning of the basal ganglia circuit. We have the frontal beta rhythm that probably reflects some local cortical mechanism. So it looked like that beta rhythm, different beta rhythm, that they have a different mechanism and they show different responses, patterns of responses to the stimuli in the task. Well, anyway, this is what neuroscience, and remember, psychiatry is becoming a branch of clinical neuroscience. So this is what neuroscience can tell us about neuronal mechanism of generation of EEG rhythm. And I have to tell you that the rhythm, as you can see from this network, the rhythm, they reflect cortical mechanism of cortical self-regulation. So the self-regulation of the primary sensory region, self-regulation of the hippocampal circuit, self-regulation, local self-regulation of the cortex. Okay, now let us see, can we use these rhythmicities to differentiate say some categories of subject. And here I present the results of wavelet analysis. So this picture shows dynamic of EEG rhythm in response to the relevant stimulus. So this is a presentation of the relevant stimulus. X-axis is time after the relevant stimulus, and y-axis is the frequency, and color depict how much the frequency changed in response to the band line. So the pink color is high increase of high level of increase, the yellow color is lower level of increase of the and on the left is the group grand average for ADHD, and on the right is healthy control. And I think that you can see here that the children with attention deficit disorder, they show a low level of frontal activation in a low beta frequency band. I think that you can see it, and basically that is why we developed the frontal beta relative beta training for ADHD to increase the beta activity and to activate in this way the frontal lobes in ADHD. Just for comparison, here I show just opposite. Here I show the group wavelet analysis for OCD, obsessive compulsive disorder patient, and the healthy control. As you can see, OCD patient, they show just opposite pattern. They show overactivation of the frontal lobe. Okay, so the amount of beta activity that is generated by the brain of this patient in response to relevant stimuli, the stimuli that have some behavioral meaning for a subject, is much higher than in healthy controls. Okay, but I have to tell you, just looking at EEG, or spontaneous EEG, even on EEG pattern in the task, gives you some information, but this information is quite limited. this information gives you a relatively limited window to the brain functioning. It doesn't tell you how well the brain processes information. It tells you how the cortex is self-regulated, and in several diseases, the cortex is dysregulated, and it's good to know when the cortex is dysregulated. But in quite many cases, the QEEG analysis, comparison of EEG spectra with the database, does not give you any clue, because there is no deviations from the reference. The EEG spectral characteristics look the same in your client as in healthy controls. So the question is, what you're going to do with this client? And to answer this question, you have actually to study event-related potentials, which is completely different from ERPs, from the spontaneous EEG, that it's evoked EEG, I mean, the potential evoked by stimuli, certain stimuli in certain task condition, and this potential, they reflect information flow in different brain systems. And here I show two systems: the sensory system, the visual system of the brain on the top, and the executive system of the brain at the bottom, which shows schematically, of course, shows brain areas that are involved in visual processing, and brain areas that are involved in cognitive control operations. Okay, the next slide. So suppose we have this queued go, no-go task that I showed you before, and we have, say, two categories of trials: go trials, two animals, animal, and the subject press a button, and the no-go trials, animal, the subject is preparing, and then he has to withhold from pressing in response to the plan, and ignore condition when the first stimulus presentation plan means that the whole trial has to be ignored. These are event-related potentials recorded in a group of healthy subjects in response to go cues. So this is a go condition, green. In response to no-go cue, no-go stimuli, red. And in response to ignore stimuli, black. And on the this is a B, figure B, and on the right there is maps they recorded at P3 waves generated by go, no-go, and ignore stimuli, and you can see that go and no-go stimuli, in contrast to ignore condition, they produces strong P3 waves, and the P3 waves for go and no-go conditions are different, have different topography, different latency. And I understand that you presume that they reflect different operation. The question is how to extract all these different operations that are happening during this task. Even just from theoretical point of view, you can say, oh, there are many things that are happening here. First of all, you have to identify stimuli, to categorize them, to distinguish plants from animals. And then you, when we're talking about the second stimulus presentation, you have to compare the stimuli that is presented to you with stimuli which is stored in the working memory, and you have to press a button if there is a correspondence between that is stored in your working memory and the stimulus presentation of withholds from pressing, and you have to detect this discrepancy to detect conflict, and then you have to suppress the prepared actions. So as I just mentioned several hypothetical operations that theoretically are happening during this task, and we developed a new methodology for extracting the component from group event-related potentials. I'm not going to talk details. We published many papers on this subject, and here I just simply present this component. Here is the component which I extracted, the tie dynamic for go, no-go stimuli. On the left, the difference wave, no-go minus go, and on the right is the topographies, I mean that the images of these components showing the localization of the component. And if you go from bottom to top, the first component represents activation of the occipital areas, activation of the dorsal stream, which is involved in categorization of stimuli. The next component is the generation of the inferior temporal lobe, which reflect the comparison of the stimuli with the trace stored in the working memory. The next component is a component that is generated in the parietal cortex, detection of the discrepancy as well as detection of the discrepancy in this area, in the anterior cingulate area, and finally the suppression of the prepared action that is associated with the activation of the pre-supplementary motor cortex. So now we know quite a lot about functional meaning of this component, and probably it's not the right place to talk about all this finding, all this research which was done in our laboratory during the last 10 years. Just to give you a flavor, in one study we recorded patients with obsessive compulsive disorder in whom the anterior cingulate cortex was destroyed. We recorded pre and post ERP. These are ERP components, and we were able to show that when you destroy the anterior cingulate cortex, only one component is decreased, and this is exactly the component that is generated in the anterior cingulate cortex, which again proved the importance of separating ERPs in the component. And then we made the next step. We asked subject to do 4 different tasks in which we changed, manipulated the stimuli. When you manipulated what kind of stimuli has to be reacted, in which the different processes like categorization, comparing to working memory, action inhibition, and conflict detection were independently manipulated. On the basis of this study, in which we recorded EEG in healthy subject and compared the behavior in different tasks, so we were able to associate some component with sensory operation, some component with action inhibition operation, and some component with the conflict detection operation. Then we ask the subject to do a battery of neuropsychological tasks. From this battery of neuropsychological tasks, we extracted parameters of energization or energizing and monitoring, and we were able to show that parameters of energizing extracted from this neuropsychological testing correlated specifically and selectively with one component, but not with the other component, and the parameters of the monitoring domain, neuropsychological domain, correlated with the amplitude of the other component. So basically, we know quite a lot about functional meaning of the components in the healthy brain, and now we can apply this knowledge to the diseased brain, to different categories, diagnostic categories. Okay, just a few words about theta-beta ratio. I think everyone knows about this ratio. There are some claim that this ratio can differentiate ADHD from healthy control. This picture shows the results of our databases. Just remind you that we have the database of healthy control. We have the database, quite large database, several hundreds of ADHD children. So on the y-axis, different age group. On the x-axis, on the y-axis, this is theta-beta ratio with standard deviations, and you can see that the ratio can indeed discriminate ADHD subject from healthy controls at early age, but not in the advanced age, not during the adolescence. And the discrimination is actually not precise. So the effect size of the difference is relatively low. Also, the first step that we try to do, we try to separate several subgroups, several patterns of spectral EEG in ADHD, and we were able to separate the group with excessive central theta, with excessive frontal midline theta, with excessive frontal beta, and with excessive alpha rhythmicities in the brain. So here I present the groups, that was derived on the basis of EEG spectra. And here I show the EEG, the grand average EEG spectra in FZ, CZ, and PZ. And one group shows excessive central theta, subgroup, the other frontal midline theta, frontal beta, and excessive alpha. So the differentiation of this group is quite helpful because it tells you what kind of treatment and what kind of problem you can see in the particular subject. But again and again, I have to stress here that when you look at the EEG spectra, you are looking at a relatively narrow window to the brain, and you can tell how the cortex in this particular subject is self-regulated. And there are subgroups of ADHD which are dysregulated, and one way of changing this dysregulation could be a neurofeedback procedure. Okay, but let us look at the other window to the brain. And here I present event-related potentials, group event-related potentials for different age groups, 7, 11, 12, 13, 14, 15, 16, 17. And that the ERP, the grand average ERP, represent ERPs to Q, the first stimulus. When you present the stimulus, the subject, the stimulus is animal, and subject is preparing to receive the second stimulus. So he keeps the stimulus in the working memory, and in this case, the parietal cortex is activated, and this activation of the parietal cortex higher in healthy controls, shown by gray color, and it's low in ADHD, shown by red color. So, this picture shows that there is at least a subgroup of ADHD subjects that show low activation of parietal cortex in response to the cues. The other, at the bottom, I show the difference wave, no go minus go. So this is a kind of cognitive control component wave. And again, it shows that the P3, the different P3 different wave is lower in ADHD in comparison to healthy control. This activity is generated in the pre-supplementary motor cortex in the front of subject. One has a parietal dysfunction, the other one has a frontal dysfunction. Okay, so if you apply the advanced method, so you separate the ERP component into the latent component, and that I'm showing here, and these are latent component of cognitive control in adult ADHD. So these are adult subject with attention deficit disorder in comparison to healthy control. So ADHD, the component are presented in red, healthy control and green, and the difference between healthy control and ADHD are presented in blue, and the p-values are presenting as vertical bars below. And as you can see, the probability of the difference is quite large, is 0.00001. So it's a high level of statistical significance. And you can see that on the bottom we have the sensory related components, which are quite normal in ADHD. Here I present in the middle, I present the parietally generated component, which is smaller in ADHD in comparison to healthy control, and on the top I presented the frontally generated component, which is higher in healthy control than in ADHD. And again, you can see that there are two different problems. One is parietally distributed problem. The other one is a frontally distributed problem. Here I present the functional neuromarkers in ADHD, the summary of functional neuromarkers showing the effect size of differences, healthy controls from ADHD for in QEEG domain. So the theta-beta ratio, frontal beta activity, frontal midline theta, mu rhythm, occipital alpha. And here I present the effect size of different ERP components. The differentiation is not bad, so the effect size is could be medium, in some cases could reach the large size. Now, I want to tell you about how this knowledge, how this information can be applied for predicting a response to psychostimuli. As you know, the first option for treatment ADHD is Ritalin psychostimulant, and you know that psychostimuli, they block dopamine reuptake it's called DET receptor in the presynaptic cleft. In the postsynaptic cleft, and it happens mostly in the striatum. This picture showing the distribution of these receptors in the striatum. And you also know better than me that in 70% of subject of patients, they don't respond to Ritalin, and the 70% response to Ritalin and 30% they don't respond to Ritalin. So there are responders and non-responders, and usually it requires 4 weeks of trial to make to the conclusion. And the conclusion is made of questionnaires made by patient, made by parents, made by teachers. On the basis of this information, the decision is made whether the subject responds to psychostimulant, he is a responder, or don't respond, he is not respondent. And well, for me it was quite obvious that to predict if the subject would respond to Ritalin or not, what you need to do is to use event-related potentials, and you don't need to wait 4 weeks. You just need actually 2 comparisons before a baseline and after 1 dose of Ritalin. Say 1 hour after taking 1 dose of Ritalin. And if you compare and you see the difference in the cognitive control component, this is a responder. If you don't see the difference, that is not a responder. The subject has not frontal, prefrontal lobe dysfunction. It has the parietal dysfunction. And this is exactly what we confirmed in our study. We recorded 87 medication-naive ADHD patients who were separated in 2 groups, responders and non-responders, on the basis of 4-week trial on stimulant medication. And we were able to demonstrate that indeed the responders, they responded to stimulant medication, and their ERPs actually normalized, they become similar to norms, while non-responders did not change these ERPs in response to no-go stimulus. Okay, so this study shows how this knowledge of ERP component can be used for predicting response to psychostimulant. The same can be done for different categories of patients, like patients with depression, patients with schizophrenia, etc., etc. Okay, so I was talking about functional neuromarkers in ADHD. Let me tell you some words about functional neuromarkers in schizophrenia. And first of all, when we analyzed the data, we collected 100 subject patients with schizophrenia. We had we have hundreds of subject healthy controls. And the first thing we did, we compared EEG spectra of these two groups. So we did not differentiate schizophrenia into different groups. We did not differentiate healthy controls in the subgroup. We just wanted to see if there is any difference, overall difference between the EEG spectra in these two groups. And basically, we were not able to see large differences. So overall, the EEG spectra don't show large effect sizes, large differences between EEG spectra. So, but in contrast, quite large differences were found, and here it is. So we found that the event-related potentials are able to discriminate the two groups, group of schizophrenic patients and group of healthy control, quite nicely with large effect sizes. Moreover, the European neuromarkers predict conversion to psychosis as well as response to antipsychotic medication. So these are the data. So these are the components that were extracted. This is a component of proactive cognitive control. So the component is generated in response to the first stimulus in the go-no-go trial, a task. Gray is healthy controls, and pink are ADHD, and green is the differences. And as you can see, p-values are quite a large differentiation. And you can see some, this component, the frontal component, do not discriminate between schizophrenia and healthy control, but these two components, which are one is pre-supplementary motor cortex component, and the other one is the temporal component, they discriminate quite nicely. These are reactive, the components of cognitive control. Again, I'm not going to tell you details, but you can see that pink lines are below the gray lines. So there is, in this case, there is decrease of the component of cognitive control in subject with schizophrenia. Okay. Now, let me just to give you a flavor. Let me show what happens in contrast to look. This is a schizophrenic patient. Now, what happens in OCD patient? Okay, this is what happens in OCD patient. As you can see, there is a completely different pattern of response in OCD patients. Instead of suppression, decrease of this component that generated in this cortical area, you can see excess of activity in the OCD patients, as well as excess of the response in the frontal component. Okay, so a few words about frontal treatment options. In 20th century, the psychiatrists have options of medication, so drugs, behavioral therapy, and electroconvulsive therapy. That's options that the psychiatrists had in the 20th century. In 21st century, psychiatry have the following new actually tools, like a neurofeedback tool, like transcranial direct current stimulation, like TMS, and deep brain stimulation. These are techniques that we are using in the clinical practice. We're using them in our clinics, as well in clinic in St. Petersburg, as well in other clinics. And just briefly, I think that most of you know the neurofeedback. Because of the time limits, I'm not going to tell you details about these techniques. Then we have transcranial direct current stimulation, in which two electrodes, anode and cathode, are placed on the head, and the current is injected. Most of the current goes through the scalp and the skin, but 10% of the current goes through the gray matter. So when the current goes through the apical dendrites of the pyramidal cell, they depolarize the membrane, and depending on the current, it could be depolarization, it could be hyperpolarization. So basically, you can either increase excitability of neuron and can decrease excitability to a neuron in the cortex. And this is exactly what is shown on this slide. This is a study that was made by Nietzsche and Pavlis when they applied anodal by red line presented and the cathodal stimulation presented by blue line, which was made on the motor cortex of the brain. And you can see that after anodal and the cathodal stimulation, there is either increase of excitability of neurons or decrease of excitability in neurons. If you make the stimulation for 3 minutes, it lasts about 5 minutes. But if you do transcranial direct current stimulation for, say, for half an hour or for an hour, it can last for hours after stopping stimulation. So it has quite long after effect. And if we are talking about schizophrenia, there was quite many studies using TDCS in schizophrenia, and this slide shows effect of TDCS on hallucination on the left and working memory in the right. And another stimulation in both cases were actually placed on the left prefrontal cortex. So the location was a little bit different, and cathode was placed in different electrodes position, but basically the left prefrontal lobe activation, another stimulation, made the difference in hallucination and in working memory, improved the working memory. The other method, which is called transcranial magnetic stimulation, TMS, which applied the Faraday law of physics, and basically the current goes through the coil. The pulse of the current, the change of the current, electrical current, induces a magnetic field, according to Faraday law, which penetrate the skull and the skin, and which generate the electric field, induced electrical field in the cortex. And if the current is strong enough, it can activate neurons locally in the cortex. So TMS used more and more often in clinical practice, and by the way, this was the first method that was approved by FDA for a treatment of depressed patient with depressions. Again and again, a left prefrontal cortex activation of the left prefrontal cortex actually helped the patients with the depression. Okay, then implementation of in the clinical practice, I follow all the time, and this is going to be the last slide that I'm going to show. How to implement this methodology into psychiatric practice. First of all, you have to record EEG, okay? And EEG has to be quite reliably recorded. So we can, using this new methodology, we can easily compensate, can correct eye movement artifact, but other types of artifact like moving of head could not be properly corrected. So the recording EEG, good recording of EEG is critical. Okay, so you record EEG. The next step would be use clinical EEG. The first thing would be finding spikes, finding a focus in the brain. And we're mostly looking at the spike slow wave complexes, which are generated by the focus, by the cortical focuses. We know from research that having a focus in the brain does not mean that you have epilepsy, but having a focus in the brain actually interferes with normal information processing in the brain, and of course can produce symptoms of some dysfunction, like symptom of ADHD if the focus is located near the Erlandic tissue. The spikes are called Erlandic spikes, and finding Erlandic spike in ADHD, it's indicated that the source, the reason for having ADHD symptoms is basically having the focus in the cortex. And in this case, I usually recommend suppression of the focus using either medication, corresponding medication, or a cathodal stimulation of transcranial direct current stimulation, or using neurofeedback. These are procedures that you can use for suppression the focus in the brain. In autistic spectrum disorder, the number of cases with the focus is even larger than in ADHD. In ADHD, numbers are about 7%, but in autistic spectrum disorder, depending again on the group, could be from 20 to 50%. So quite large percentage. The next step, of course, is to compute EEG spectra in eyes open, eyes closed in the task condition, and compare with the spectra. It gives you information how the cortex is regulated in this particular subject. And in this analysis, you rely on the data that are known, say on the theta-beta ratio, on excessive frontal midline theta, on excessive alpha activity. So you rely on the knowledge that's already known, and your job is to define what type of cortical cell regulation is present in this particular subject. If it corresponds to the symptom the subject is presenting, and the ways how the cell regulation can be corrected, can be learned how to deal with this. And the neurofeedback in this case is definitely a leading method of dealing with these cortical dysregulations. The next step is to compute event-related potentials in the task. Depending, again, the task has to be selected on the basis of the problem. If the problem lies in the cognitive control domain, then you select the go, no-go task. And then you can select different components from event-related potentials, and you can say what component is impaired. In this particular subject, you know the localization of the component, so you can use either neurofeedback techniques to work on this area, or using the transcranial direct current stimulation, or using TMS, or if you know that this component is the one that responds to the particular medication, then you can use the corresponding medication. Okay, this basically wants a kind of a theory that can be applied in clinical practice.